that does work. Cool. First of all, I'd just like to thank the organizers for this great opportunity to present some of the research we've been doing in our lab. I'm going to be talking about a project an extended project we've been working on using stable isotope probing to look at soil microorganisms and their involvement in soil carbon cycling. And this project uses both 16S sequencing as well as metagenomics with assistance from the JGI. So most of this introduction, you've seen much of this before in Dr. Firestone's talk. So soils have a large stock of carbon. It's estimated in the top 3 meters of soil, there's over 2,300 petograms of organic carbon. And this organic carbon is very important because it's relatively bioavailable and therefore plays a large role in the global carbon cycle. And microbes are a key component of this system. They're important for both the input and output of this organic carbon through breaking down complex organic matter like leaf litter, and loss of this organic carbon through soil respiration. They're also essential for the turnover of this organic carbon within the soil and likely involved in stabilization and reacquisition as well. So with more and more studies, we're learning more about these organisms, but unfortunately, despite their vast diversity, this diversity is not really reflected in the culture collection. And so there's still questions that we have based on their ecology and their life history strategies and how these factors influence their role in carbon cycling. So to go about this, we use a method called DNA stable isotope probing. So this allows us to identify microbes that metabolize a given substrate. So in a basic SIP experiment, we have an environmental sample. In our case, this is a soil microcosm. We then add a 13C labeled substrate. So this could be glucose or cellulose, where all of the carbon atoms are the stable isotope 13C rather than 12C. The cells within the environmental sample that metabolize this carbon will incorporate that 13C into their DNA. So when we extract DNA, we have this mix of 13C labeled DNA from these incorporators, as well as just the regular 12C DNA from the rest of the community. We then use ultracentrifugation with a cesium chloride gradient to look for a shift in buoyant density of this labeled heavier DNA compared to its 12C counterpart. So for this initial study, which was done by Nick Youngblood and Chantal Coakley, they took 9 substrates, which are representative of what you might find in plant biomass. They mixed these 9 substrates together as a simulated plant biomass additive and added this to all of the soil microcosms. These microcosms were split into 2 groups. There's a 13C treatment group, where one of these substrates is labeled with 13C, while the other 8 substrates are unlabeled. There's also the 12C control, where all 9 substrates are just 12C unlabeled. They then extracted DNA from all these microcosms over multiple sampling time points, fractionated this DNA, sequenced the 16S ribosomal RNA gene from each fraction, and identified the operational taxonomic units that were significantly labeled with 13C in 1 or more treatments. So out of a total of 4,800 OTUs, over 1,200 of them were labeled with 13C in 1 or more treatments. Don't have to pay too much attention to this heat map because it's quite complex, but this just shows you the incorporation pattern of all of these incorporator OTUs. And you can see that there's a great diversity of these incorporators. So to get a better handle on this data, they grouped these incorporator OTUs into 28 functional guilds. And these guilds were based on that incorporation pattern that was in the previous figure. So this is just a heat map showing the incorporation pattern of these different functional guilds, where you have functional guild just numbered on the y-axis, and then the substrate and day on the x-axis. And the color indicates the number of incorporators within that functional guild that were labeled on that given day. So for example, these functional guilds down here on the bottom are incorporating more insoluble substrates like cellulose and palmitic acid, and do so very late in the experiment. These functional guilds up on the top, however, are incorporating more soluble substrates like glucose, xylose, amino acids, and tend to be incorporated very early in the experiment. So now that we have these functional guilds, they wanted to look to see if there was any life history strategies that can be observed in them. So they ran a principal component analysis with all 28 functional guilds. And this analysis was based on the number of substrates incorporated by each guild, the relative abundance of the member OTUs in the bulk community, an estimate of the ribosomal RNA copy number of these different OTUs, a proxy for the bioavailability of the substrates that labeled these OTUs, and then the time point of first incorporation, so how early were these OTUs labeled. And so the main thing that you can see from this principal component analysis is this delineation of these functional guilds along what we're calling this copiotrophic to oligotrophic gradient, which is essentially this principal component 1 axis. So these more copiotrophic functional guilds are labeled by more soluble substrates, are labeled early in the experiment, tend to have high ribosomal RNA copy numbers, and also tend to have a high relative abundance in the bulk community. These more oligotrophic incorporators, functional guilds, on the other hand, are pretty much the exact opposite. They are incorporating more insoluble substrates and tend to be labeled very late in the experiment. So now that we have this idea of different life history strategies of these active organisms, and we know their function, we want to be able to link this to the genomes. So we want to ask questions related to their metabolic capability, their auxotrophies, production of various secondary metabolites, and motility and cell attachment, as well as a few others. And to do this, I'm going to use metagenomics. So I specifically chose 8 of these treatment samples that I showed you before, and I chose these to target these labeled organisms from these functional guilds. And specifically, I chose them because they had a high relative abundance of these labeled organisms. So here on the y-axis, you just have the relative abundance of labeled OTUs in each of the samples. And so you can see in all samples, we have over 5% of the community consisting of these labeled OTUs. And you can actually see the trend of which types of organisms we're targeting here, where these earlier samples that are treated with glucose, xylose, and glycerol are targeting more copiotrophic incorporators, while these later samples with palmitic acid, cellulose, and vanillin were targeting more oligotrophic incorporators. So for this experiment, I'm also using CYP-enhanced metagenomics. And so by this, I mean that I'm taking the heavy buoyant density window of that cesium chloride gradient for each of these samples. And so by choosing this heavy window, I did not only choose samples that have a high relative abundance of these target organisms, but now I am enriching for the labeled DNA of these organisms. So with the help of JGI, we have sequenced all these samples. Currently, we are running a co-assembly and binning the metagenome assembled genomes. Once these MAGs are complete, we will combine these results with our 16S CYP results that I showed you before to assess the genomic signatures of different life history strategies. As I said, this is still ongoing. So in the next couple slides, I'm going to show you some very preliminary data based on individual assemblies and annotation and analysis with IMG. And so for this analysis, I'm looking for enrichment of genes or gene categories due to 13C. So here I'm comparing the 13C treatment samples with the corresponding 12C control sample. So that if there's an enrichment of this gene or category, it suggests that it's found in genomes that are labeled with 13C. So the first thing I looked at was to see if I was enriching for any carbohydrate active enzymes. So I took KEGG orthologs annotated by IMG and mapped them to glycosyl hydrolases from the KZ database. And so here I'm just showing you the number of these glycosyl hydrolases that are enriched in the treatment sample compared to the corresponding control sample. And the main thing you can see here is, first of all, we are enriching for these glycosyl hydrolases, which is important because we believe these enzymes are important for breakdown of plant biomass. And you can even see a trend where the palmitic acid samples and the cellulose samples have more of these enriched glycosyl hydrolases. And largely, this seems to be driven by these enzymes targeting oligosaccharides and cellulose. Interestingly, we don't see much enrichment of these glycosyl hydrolases in the vanillin sample, despite the fact that we do believe that we are enriching for these oligotrophs. And so I believe that this really suggests that there's just a great diversity of metabolisms of these oligotrophs. So the other thing I looked at was to see if there's any gene categories that might be associated with different life history strategies based on their enrichment patterns across samples. So here I'm using a variable that I'm calling the weighted average of that principal component 1 axis I showed you before. And so this essentially just stands for the dominant life history strategy of the incorporators within each of the different samples. So everything on the right-hand side of the figure in the positive region are samples with more copiotrophic incorporators, while everything on the left-hand side in the negative region are samples with more oligotrophic incorporators. And so these first two examples of COG categories seem to be associated with a more copiotrophic lifestyle. So amino acid transport and metabolism and carbohydrate transport and metabolism have a greater enrichment in samples with more copiotrophic incorporators and little to no enrichment in samples with more oligotrophic incorporators. On the other hand, lipid transport metabolism and post-translational modification, protein turnover, and chaperones seem to be enriched in samples with more oligotrophic incorporators and might be a sign of that life history strategy. And so, as I said, this project is still ongoing, but I believe that this preliminary data shows that we are in fact enriching for different genomes based on life history strategy. And hopefully when we get these mags back, we'll be able to study that a bit more. And with that, I'd thank everybody in the Buckley Lab group and the JGI who've worked on this project, as well as my funding sources. And if you have any other questions that I can't answer right now, please stop by my poster. It's number 51 tonight. And yeah.
This may be a naive question, but how do you differentiate when the labeled compound is actually being metabolized directly by one organism? Because then you could have another organism that is feeding on the first organism that assimilated glucose, for example.
Yeah, that's a great question. So I think we can't necessarily say that the secondary feeding is not occurring. I believe, though, that the time points that we chose, so the early time points for like the glucose and xylose, I think that we're still mostly capturing these primary feeders. And same thing with the later sampling time points for those more insoluble substrates. But yes, that is a possibility that might be occurring.
I want to ask one. So we've been working on trying to automate this process. It's incredibly labor intensive. And so you've labeled with, I think it was 9 different substrates at multiple time points. Do you have any kind of automation to do that? Or was that just how many person years did that take to do all of those separations?
Yeah, so for the actual cesium chloride gradients, that was all just done by hand.
Yeah.
So
wow.
we spend all morning doing the fractionation. But for like for PCR and that part, we do have a robot that is doing that. So at least that part is done.
Yeah.
Automated.
With the fractionation.
Yeah.
All right, great. Well, thanks again for a great talk.